The overall goal of this procedure is to generate cortical interneuron progenitors and post-mitotic interneuron precursors from mouse embryonic stem cells using a modified embryoid body-to-monolayer method.
This method can help answer key questions in the neuroscience field, such as how subtypes of cortical interneurons achieve their individual fates during development. The main advantage of this technique is that it enables efficient generation of NKX 2.1 expressing interneuron progenitors, as well as methods to enrich for parvalbumin or somatostatin interneuron subgroups.
To begin this procedure, plate mitotically inactive MEF feeder cells in MEF media onto tissue culture-treated plates. Allow at least 12 hours for them to settle in a cell culture incubator at 37 degrees Celsius before plating the ESCs. If not used immediately, replace the MEF media every 3 days. Next, add ESCs containing mouse leukemia inhibitory factor to the MEF plates. Incubate the cells at 37 degrees Celsius and 5% carbon dioxide. Passage ESCs using trypsin EDTA once the dish becomes 70 to 80% confluent, and maintain the ESCs on a MEF feeder layer in ESC media to keep pluripotency. Before starting differentiation, passage the cells at least once onto gelatin-coated plates without MEFs to dilute them out. To do so, prepare gelatin-coated plates by adding 0.1% gelatin and PBS with calcium and magnesium to a tissue culture-treated dish and incubate at 37 degrees Celsius for at least 1 hour.